Yeah,
Thank you
thank you,
very much.
Alex, for that wonderful introduction. We are delighted to contribute to this discussion about technologies in the lab. The first slide you see there, that is me and Dewi interacting with a protein. So we worked with an amazing group at Curtin University to design VR-based learning activities and to employ them in first and second year chemistry labs. So what we are going to share today is how students were collaborating to learn enzyme-substrate interactions in virtual reality and what kind of learning they got. We are going to co-present, so Dewi is in the control center. She will be moving the slides. As I mentioned, we worked with the team. We're working on an ARC-funded project, which is about using VR for helping students to visualize abstract concepts. As you can see, we had our team leader was Associate Professor Mihe Won. We have other members, Professor David Triggers, Professor Moro Moserino, Chin Chang Chai, Professor Roy Tasker. Alex, you know this whole team. So yeah, this is the team that we worked with. Now we are looking at enzyme substrate interactions because the concept is really fundamental in both chemistry and you can say biochemistry or even biology. So a good understanding of this concept helps students interpret catalysis, drug binding, or inhibition. But the challenge is visualizing protein structures is really hard because we are dealing with very complex molecules here. We have a few structures. When you look at the diagram, it is really hard to appreciate the complexity of this kind of structure. You look at the different amino acid residues. It may be trivial for a chemist who is experienced, but for students, they find it really hard to understand the scale, the depth, and the 3D nature of this kind of structure. When we teach, we go for, sometimes we use diagrams, sometimes we use analogies, lock and key, or sometimes we use practical work. But all these kind of strategies have their limitations. For instance, if we ask students to go to the lab and try out an experiment with enzymes, they could end up generating a bunch of numbers, but interpreting what these numbers mean in relation to what is actually happening at the molecular level is a bit challenging. Now, research has shown that when students fail to visualize or to think at the molecular level, they tend to rely on rote memorization. They can repeat the facts, but understanding or explaining what it actually means is a bit of a struggle. So our team is using immersive virtual reality. VR has been hyped for its potential to show 3D structures. You can also manipulate the structures. You can play around to see what works and what does not work. For some programs, students can actually collaborate with their peers to share ideas, and we all know the benefits of collaborative learning. But when we did a literature review on how IVR was being used in education, in science education, many researchers were focusing on engagement. Students would say, wow, this was amazing. This is really good. I felt very engaged. But we also found that there were very, very few studies that were focusing on what students were actually learning. We also found that there were few studies that were going into exploring the potential of VR for students' collaboration. So it was mostly individual students playing with the tool. So we designed this study to address the two gaps. One, to look at what students actually learn, and then to look at what students do within VR. How do they interact, one with themselves, with the peers, and also with the molecular structures. So this is what we're addressing in this study. Two research questions. One, what is the level of undergraduate students' understanding of enzyme-substrate interactions before and after a VR activity? And then two, how do pairs of undergraduate students interact within the virtual reality environment to learn the concept of enzyme-substrate interactions? Dewi will start from here.
Okay, so thanks, Henry. When we look at the literature, we found that there's many IVR applications out there for chemistry, but mostly they are 360-degree visualization or virtual lab, and they're mostly single player. For this study, our project, we have in-house program. So we have this full body interactivities. Two students can walk around the room freely with the observer outside their space. And it's a collaborative mode. So two students sharing the same virtual space here. They see what the other can see. And this VR application that we use for this study has a series of tasks. So first, they explore the protein enzyme structure. There's a different type of visualization they can explore, the ribbon, the electron density, and discuss about that. And the second one is placing the substrate at the entrance of the enzyme. So they have to figure out the best position for it to react. And then lastly, they can explore the enzyme catalytic chamber. So actually go inside the enzyme and see how it looks like and where the everything's are positioned. So for the data collection, we have 42 pairs of undergraduate students enrolled in the chemistry units. And we record their audio, what they say, and the video. And we transcribe them and create diagrams for them. I will go into that later on in more detail. So before and after the VR activity, we have pre-interview and post-interview, approximately 20 minutes, and the VR activity itself is 25 to 40 minutes, depending on the pair of students itself. So the interview mainly have two different parts. The learning part is we ask them, what is your mental image of an enzyme? Describe it to us. And then not only describe, we ask them to draw and then explain the drawing and then explain why do you think enzyme reaction is very fast. So we want to dig in what they learn about enzyme. And of course, the overall experience itself, how was it? And if they say fun or not fun, want to know why. So the data analysis here, we have divided into two research questions. The first one is the conceptual changes about the learning before and after IVR. We have a student-generated diagram, their explanation of the diagram, and conducted a multimodal inductive analysis of it. Here you can see on the right side is an example of what students draw. So we have 18 pairs who exhibited some understanding of that enzyme-substrate interactions. And for the second research questions is dealing with what actually happened inside the IVR. We have a case study, multimodal cross-case analysis. So the video itself, we created a synchronized video of the room. So we see what the student do and what each student see. And from that video, we created a location map to kind of follow how they move inside the VR space. So we draw how the enzyme is and the direction side and the position of each student and the positions of the substrate that they move around. So the results. The learning first, we'll go to Henry, please.
Yeah. So our first research question was exploring what do students actually learn from VR. So one of the questions we asked in the pre-interview was, what does an enzyme look like? Now, surprisingly, even though some of these students were doing biological chemistry or they had very good chemistry backgrounds, they had very abstract ideas of what enzymes looked like. Some were mentioning that enzymes are proteins, they are made of amino acids, but when you actually ask them further, they were not sure what it looked like. For example, Albert here was saying, I don't remember seeing a proper one, but I think of it as a virus, COVID. So That is the idea. Others had a similar idea of a ball. They would describe it as a ball. When we asked them how enzymes interacted with substrates and asked them to draw, they were giving very simplistic models of enzyme-substrate interactions. For example, the famous lock and key. They would explain you have the enzyme, you have the right shape of the substrate, and there you go, you have the products. But when we would ask them what is actually happening here, they're like, I'm not sure, but I know this is what is happening. We also gave them an example of to try to put the rates in context. Would tell them, you have an enzyme, you have a substrate, the rate is going to be very fast, maybe 10,000 molecules per second of the substrate being broken down. But in the lab, if you carried out the same hydrolysis reaction, it is going to take hours. Why do you think that is the case? Most of the students were unsure. They didn't know. They were like, I know it happens. I know the enzyme reaction is fast, but I'm not sure why. Others would explain using activation energy. They would say the enzyme provides an alternative pathway with a low activation energy. But when we would ask why, what is it about the enzyme that gives this reaction a low activation energy? They were not sure. Jonah says, it basically short circuits the reaction, but I'm not sure how. Others would imagine the enzyme holding the substrate for the substrate molecule to wait for other molecules to react with it, but they would not explain whether it is electron density or what exactly was happening. So they simply had hypotheses of what was going on. That was before VR. Now, after IVR, we asked them, what did you learn about enzymes? Students realized that it was not just about the shape aspect that they were highlighting in the pre-interviews. They realized the scale of the protein. They realized the role of electron density. They realized the importance of the catalytic triad. Ellen here had drawn this beautiful lock and key model, but she said, I knew there were lectures about the catalytic triad, but I did not know. It was really hard for me to visualize it. But after IVR, she understood what was going on. For example, here she critiqued the simplicity of the 2D diagram, the lock and key diagram. She says, this is definitely not what happens. This is very simple. Yes, but it shows that there is the particular shape, but that is not all that is happening. Now, we also asked them about the rate of the reaction. Now that you have done the VR activity, why do you think the enzyme reaction is very fast compared to a lab reaction? Students started integrating multiple concepts into their explanations. For example, the relevant amino acids that are taking part, the role of the orientation of the substrate or role of electron density, and the collision theory. They would think about a lab reaction where you need to rely on chance, random collisions, and then they would contrast that with an enzyme structure where everything is in the right orientation with electron density and everything is ready to go. Some students would refer to the enzyme as a machine or as a factory to really explain the efficiency of the enzyme. So what we found was by interacting with the models in VR and trying things and pushing the molecule through, orienting it in the catalytic chamber for the reaction to take place, students got a very concrete idea of what an enzyme reaction looked like. There, your mic is off.
Sorry. Okay, thanks, Henry. So we heard about what the students learned before and after IVR. So now it's what they actually do. We found that there's two distinct pattern of student interaction inside IVR. We go for the first one is Betsy and Owen interactions. So this pair of students exhibited a discussion. They discussed with each other, but the discussion was dominated by Owen, the male. And we can see here in the location map that this is the bird eye view of the VR space. We can see that Betsy and Owen are mostly standing at the entrance of the enzyme. They just push the substrate at the entrance of the enzyme there, trying to match the blue and red. So the concepts they're discussing between them is mostly only about electron density. So in the end, the answer about what's the best way to place the substrate in the entrance of the enzyme was incorrect. They place it like this instead of like that. And let's see the second type of patterns. So we here have Maya and Ben. Again, they both friends and male and female, but they have a more collaborative discussion between them. So Maya would have an idea, Ben would refute them and give another idea. And then we noticed that they try to push the substrate into the enzyme and then pull it back and trying to find the best way to for it to position to enter the enzyme. So it's we can see they're more explorative than the previous pair, Betsy and Owen. They even go inside the enzyme itself with the substrate. So the concept they able to discuss is electron density and the shapes of it and the mechanism of reactions. And in the end, their final answer was correct. This is the best position for substrate to enter the enzyme. So what is the significance of this study? So in the beginning, we know about students' conceptual change. The research gap is there's an interest in IVR. VR is cool for education, but the learning benefits itself was not clear. So we know that realistic looking 3D enzyme environment actually improves students' understanding. But we have to carefully design the IVR application towards what kind of learning goal and what kind of challenging concept that we want to them to learn. And the second one is the students' interaction. Oh, yes, we know that IVR can be exciting for students, positive for engagement, but how they use it, it's really not clear yet. So by looking into what the students do inside IVR, we know there's some kind of influencing about how they walk around and how they collaborate with each other to what kind of concept they able to discuss and learn. Oh, and so practical implication, we have the first one, evaluate. So we kind of have to consider the alternative ways. So maybe multiple choice questions is good, but asking them to draw will benefit for us to know what they can learn better. And the benefits of VR is realized differently, depend on what kind of students they pair with and their background. So we need to be careful with that. And the unique but educational benefits of IVR, we just also need to be mindful for that. So we don't just use the same learning way for each media learning approach for media, all media. And that's it. Thank you.